Hello students and welcome to our next lecture of Path 231. Now we are going to see the morphological characters of bacteria. So there are different kind of pathogenic bacteria which we are going to study in plant pathology. But there are some of the common characters which found in the bacteria on which basis we can be classify or we can identify a proper bacterial function and bacterial classification and its characterization. On that basis, these are some of the common morphological characters which are going to see we are now. Number one, no nucleus. Unlike other eukaryotic cell, bacterial cell is the only cell which lacks prominent nucleus. In case of fungus, as we have seen that it is eukaryotic in nature, is well defined nucleus is there, but in case of bacteria there is no well defined nucleus, so it is known as prokaryotes. So that's why there is no well defined nucleus. That is the basic first character in the bacteria. Second, presence of cell wall. Bacteria has a cell wall which is made up of different substances like glycoproteins, lipopolysaccharides and the lipoproteins. So basically the cell wall is there in case of the bacteria and having basic substances like glycoproteins, lipoproteins, lipopolysaccharides and the lipoproteins. So in case of this diagram, you can see here the bacterial cell or outer membrane and as well as the particular peptidoglycan and the periplasm outside the cell wall and in between the outer membrane and the capsule. So this is about some of the components which are present in the cell wall. Then cell membrane. This is present immediately below the cell wall and outer membrane called periplasm, which lies below cell wall but above the cell membrane. So the periplasm you can see in the picture, that is periplasm. So the area that is the periplasm, particularly known as in between the cell wall and the inner membrane. So that are the main component of the bacterial cell in between the cell wall and the periplasm. So cell membrane, that is the particular called as a periplasm. Next is a pili. These are small and tube-like projection from bacterial surface. They are the organelles of sexual reproduction and are involved in exchange of genetic material as a part of reproduction between two bacteria. You can see here the fimbriae or a pili, known as a projection hair-like structure, which is important for the transfer of genetic material during the reproduction. Particularly, these pili are present at any stage or any the part of the bacterial cell in the form of hairy structure. These pili are projection, extra projection from the bacterial surface, and then majorly plays a role in the sexual reproduction for the exchange of the genetic material. Now, fifth point related with the characters is food dependence. Bacteria majorly depends on the synthesis of its own food. There are so many photosynthetic bacteria are there. There are so many autotrophic, heterotrophic bacteria are there. So all these bacteria are synthesize their own food in different ways. So they are selfly food dependent bacteria. In case of plant pathology, there are also some autotrophics are there, some heterotrophics are there, and photosynthetic synthetic bacteria is also there. Now, sixth character is reproduction. Most of the organisms have one or two methods of reproduction, but there are multiple ways of reproduction in bacteria. But majorly, which are important in our plant pathology, that is asexual reproduction, sexual reproduction, and there is various method, that is vegetative reproduction also. But in pathology, we have only the asexual and sexual reproduction is important. Asexual reproduction, the bacteria cell undergoes to produce two bacteria by binary fission and also forms the endospores. This is asexual reproduction. One septation develops in adult bacteria to form, and also the separation of the nucleus, separation of the genetic material into two daughter cells. So it is known to be the particular mode of action or the method in the binary fission, and also formation of the endospore. These are the two methods of asexual reproduction in bacteria. Second, that is sexual reproduction. It occurs by method like transformation, conjugation, and transduction. These are three methods. We are going to see the detailed reproduction of bacteria in detail in the next lecture. So I am going to explain you this in detail by showing the images and some photo images as well as videos in the next lecture. So these are the sexual reproduction, transformation, conjugation, and transduction. Seventh point about the bacteria, that is spore formation. When the environmental conditions around are not suitable, the bacteria converts to a hard spore form. This spore is highly resistant to heat, chemicals, and drought conditions. There are so many bacteria which develops the spores when there are some adverse climatic condition like heavy temperature or maybe some of the factors which are not suitable, when there is no any moisture or maybe precipitation is heavy, there is heavy humidity is less. So these are some environmental conditions which are not suitable for the development of the bacteria. So these bacteria perpetuate in the form of spores. They converts themselves into a hard spore structure, that is a special character in the bacteria to survive for a longer duration in any such kind of habitat, maybe soil, maybe water, maybe plant debris, maybe anything, plant parts. So it is in the form of the spore. So the spores are highly resistant to heat, chemicals. If any heavy temperature or chemicals comes in contact with the spore, so there should not be any adverse condition in the genetical behavior of that particular spore. So from the spore there is a further development of the bacteria maybe occurs in the next season. So this is a special character in the bacteria which is found unique, that is known as a spore formation. So moving towards the special character, that is absence of the cell organelles. Other cell organelles like mitochondria, Golgi bodies, endoplasmic reticulum are absent in the bacterial cell. That we have seen in some other cells like fungal cells. There are some of the cell organelles are present. So liposomes, lysosomes, these are so many factors which are to be absent. But you can see here in the picture, ribosomes are there, the mesosomes are there. So, but most of all, cell organelles like mitochondria, Golgi bodies, and endoplasmic reticulum is absent in the bacterial cell. Now, next is flagella are present. Some of the bacteria are motile, especially the bacillus type having the flagellation. So you can see here at the corner, the big hair-like structure that is known as flagella. Most of all bacteria, and they have motile, means they can swim by the way of this flagellation from one place to another place, means the locomotion is there, so motility is there to cause the infection to the other plants or other part of the soil. So especially the bacillus type of bacteria having the flagellation, it's a very unique character in this the bacterial cell. Now about the next character, that is bacteria shape. These bacterial structures have specific shapes like stick, spherical, comma, spiral, or in a bunch or isolated. Particularly in the case of stick, that is bacilli, bacillus shape. Spherical means focus. Comma shape, that is the cholera bacteria is having the shape that is comma shape. And spiral, that is known as spirochaete, or in a bunch form they can be formed, or isolated, only one single colony can be formed of the bacterial shape. So these are separate shapes, different shapes of the bacteria can be found in the vicinity of the water, soil, or maybe on the plant parts. The habitat of the bacteria, next character, the bacteria are the only organism which shows cosmopolitan distribution. They are present in air, water, soil, snow, etc. Everywhere, means habitat of the bacteria is everywhere. So that's the life of the human beings. It is we have seen so many videos in the YouTube that it starts from the bacteria. So starts from the sea bacteria. So the bacteria can be seen in at anywhere, maybe on from the Arctic to Antarctic to anywhere in the sea and or in the soil. So the bacterial structure is in a such a way they can modify it as per their habitats. Means maybe it is heavy particle soil, if it is heavy water, maybe water from the rivers, maybe from the stagnant water, maybe from the running water, or maybe in the sea water. So then there should not be a different kinds of bacteria can be seen. So everywhere the habitat of the bacteria all over the world. Now last point of the character, resistance and tolerance. Some of the bacteria are highly resistant to adverse environments. Even they get tolerance to harsh chemicals and other stuff which destroy them. So it's having some kind of ability of the bacteria to resist and tolerate to adverse climatic condition like heavy temperature. Some of the bacteria can resist to heavy temperature up till 45 degrees centigrade in the soil also, in case of our plant pathology. And some of the bacteria are also very resistant to some chemicals, means we can call it as antibiotics. So they can't, the antibiotic can't kill the bacteria. So these are some of the ability of the bacteria. They can develop amongst themselves to cause the resistance and the tolerance due to environment and the chemicals. So these are some of the general characters, basically the morphological character of bacteria. So these are the 12 points which you have to write in the examination, or the important question for your end exams, theory exam particularly, an important topic in your examination for the syllabus theory examination. So you have to write maybe a 4-mark question, maybe ask for morphological characters of bacteria. Now, next small topic about the bacteria in relation to plant pathology, the classification of the phytopathogenic bacteria. Phytopathogenic, which are causing the infection or a disease to our plants, that is crop plants. So these are the classification which is related with the phytopathogenic bacteria. The 3 divisions are there in case of this classification of the bacteria, phytopathogenic bacteria. The 3 divisions are: first division, Gracillicutes, that is mostly known as gram negative bacteria. Second division, Firmicutes, that is gram positive bacteria. And the third division is Tenericutes, that is known as a Mollicutes group, that is which include the phytoplasm. One by one we are going to study about the divisions. First is Gracillicutes, known as a gram-negative bacteria. Class, Protobacteria. Family, Enterobacteriaceae. And the genera, 3 genera are there: Erwinia, Serratia, and Spingomonas. So these are some of the important bacteria in which genera Erwinia amylovora, which causing the fire blight of apple. So that are the important disease in case of the bacterial pathogenic disease survives or maybe spread all over to the world, important disease. So it comes under this Gracillicutes. Next family under division Gracillicute, that is Pseudomonaceae. Genera Pseudomonas, Acidovorax, Ralstonia, Rhizobacter, Xanthomonas, Rhizomonas, and Xylophilus. This is the important family which is in relation to our plant pathology and plant disease. And important bacteria are Pseudomonas and Xanthomonas and Ralstonia. Basically, Pseudomonas and Xanthomonas are the most important, which are causing the majority of the disease in our plant pathology. So these are some of the genera, having 7 different genera, out of which Pseudomonas, Xanthomonas, and Ralstonia are important in this family, Pseudomonaceae. Next family under Gracillicute, that is Rhizobiaceae. Genera are Agrobacterium and Rhizobium. In case of family Rhizobiaceae, first is Agrobacterium, that is the pathogenic, or we can call it as a harmful bacteria, Agrobacterium tumefaciens, that causing the galls on the roots of the plant. And second is Rhizobium. Rhizobium is a beneficial bacteria. It's known as a nitrogen fixing bacteria, which comes under the family Rhizobiaceae. And family last, it is unnamed family, that is genera known as Xylella and Candidatus. So these are about the division Gracillicutes, that is known as a gram-negative bacteria. Mostly these are covering the plant pathological diseases or plant diseases which are important in our plant pathology like Erwinia, Pseudomonas, Ralstonia, Xanthomonas, Agrobacterium. Okay, so now we are moving towards the second division of plant pathogenic bacteria, that is Firmicutes, basically known as gram-positive bacteria. Class Firmibacteria, the genera are Bacillus and Clostridium. Okay, and in case of this Bacillus and Clostridium, so most of all, very rarely the bacterium are causing the infection in case of plant pathology. The then second class is Thalobacteria, so known as a branching bacteria. There is no any important genus or genera important in this Thalobacteria. The basically some of the relation with, not in relation with the plant pathology. These are number one, Arthrobacter, Clavibacter, Curtobacterium, Lephosonia, Rhodococcus, and Streptomyces. Streptomyces is having a filamentous bacteria or branching bacteria. Streptomyces cabees, only the genera having important for the scab of potato and scab of apple. So in our plant pathology. So there are the two classes, Firmibacteria and Thalobacteria. It's important in the division Firmicutes, known as a gram-positive bacteria. Third division in Tenericutes, that is Mollicutes. Class Mollicutes, family Spiroplasmataceae, genus Spiroplasma, family others, second family that is unknown family, and the genus is Phytoplasma. Phytoplasma are also known as MLOs, mycoplasma-like organism, so which comes under the Mollicutes and having a different genetic misbehavior in the plant to cause some typical diseases like little leaf of brinjal and some grassy shoot of sugarcane. So these are some of the diseases which are causing by the Phytoplasma. So these are the three important divisions: Gracillicutes, Firmicutes, and Tenericutes, which are important in case of the classification of phytopathogenic bacteria. So this is your important topic which we have covered today, relation with the morphological characters of bacteria. 12 important characters are there, and the classification of phytopathogenic bacteria, that is Gracillicutes, Firmicutes, and Tenericutes. Maybe the question arises like this, write in detail about the morphological characters of bacteria and along with the classification of phytopathogenic bacteria in detail in the final examination, maybe for 4 marks. So this is about the today's lecture. In the next lecture, we are going to see about the vegetative, sexual, and sexual reproduction in bacteria. So we are dealing with, are going to study the details of reproduction in bacteria in the next lecture. So see you soon for the next lecture. Good day.